is Céline Scornavacca from the Institute of Evolutionary Sciences of Montpellier. She's going to talk about the Multilocus multispecies coalescent, a flexible new model of gene family evolution. Céline is not physically here virtually here in Zoom today. We have her video, and then she will join us by Skype to answer your questions. So Jesus, feel free to start with the video when you're ready.
Okay, let me check if the sound is okay,
Scornavacca, and today I'm going to present a new model of gene family evolution that my collaborator and I call the Multilocus multispecies coalescent. So there are two important kinds of phylogenetic trees which are keys to the study of gene and genome evolution, and those are the gene and the species tree. Species tree depicts the evolutionary history of a set of organisms, while gene trees depicts the evolutionary history of a set of homologous sequences. And in the species tree, we have that the nodes represent speciation events and branch lengths represent the divergence time and substitution per site, while in gene trees, nodes reflect different processes in addition to speciation, such as variation in gene copy number or gene transfer between species. When gene tree reconstruction are performed, one often observes that gene trees disagree with the species tree. Such a discordance can have two main causes. Either it is due to inferential error or model inadequacy, that is, the sequences involved according to a process violating the assumption of the model used to infer the tree, or to events in genome evolution that leads to topological conflict between individual gene trees and the species tree. These evolutionary processes can include several gene range events, such as gene duplication, gene losses, and horizontal gene transfers. In a gene duplication, a single gene copy gives rise to two copies at two different loci, the original locus and a new one. In a gene transfer, a gene from one species enters the genome of another contemporary species. Transfer can occur frequently, for example, in bacteria, and through a number of biological mechanisms, such as transformation, transaction, and conjugation. And in a gene loss, a gene is simply removed from the genome. Collectively, we refer to these processes as DTL, and this event can occur multiple times, allowing the gene tree to possibly differ greatly from the species tree. Another process that is considered in this work is incomplete lineage sorting, or ILS. ILS is due to polymorphism, so having different alleles in the same population, that is maintained through successive speciation. In our example, we started with a population containing a single white allele, then a mutation creates a black allele, so we have polymorphism in the population. Then we have a first speciation that takes place, rapidly followed by a second one. As the white and black alleles still coexist when the second speciation takes place, then both alleles may be fixed in the separate descendant species. In our example, the black allele is fixed in the rightmost population, while the remaining have the white one. This can produce discrepancy between the gene tree and the species tree. Hemiplasty is a term used to refer to the species tree gene tree conflicts that result from incomplete lineage sorting. It is important to realize that such a scenario is entirely neutral. There is no need to assume differences in the fitness between the white and black alleles for such an occurrence to occur. Coalescent theory provides a genealogical interpretation of ILS that helps connecting this phenomenon to gene tree species tree discordance. The key point is that the age of the common ancestor of two gene copies sampled in two different species is older, in absence to DTL events, than the time of the speciation between the two species, because of polymorphism in the ancestral species. When the speciation occurs far apart in time from each other, ancestral polymorphism only creates differences in branch lengths. If, however, two or more speciation occurs in a time interval of the order of coalescence time, then the coalescent and speciation may interact. This can cause not only branch lengths, but also topologies to differ. ILS and the multispecies coalescent have received much attention after the discovery that classical phylogenetic methods are inconsistent in a subset of the parameter space and in the context of the study of convergent evolution. So far, ILS has been mainly considered separately from other sources of conflict between species tree and gene trees. However, it can interact in complex ways with the process of gene duplication, loss, and transfer, as first noted by Rasmussen and Callis in 2012. It is because DTL events spend some time in a polymorphic stage in a population before they become fixed. Thus, individuals can differ from each other in terms of gene copy number. Here we have an example of ILS interacting with loss. With only one loss event, two descendant species may end up with an empty locus due to the presence of ILS, since an allele which is not yet fixed is lost. Here we have an example with duplication where a newly created locus does not fix in the old descendant species. So the duplicated gene only fixed in species A and B, and species C ends up with only one copy, but without the need of gene loss events. We call the discrepancies between the gene copy number that result from the interaction between ILS and DTL as a copy number hemiplasty, or CNH for short. So there are a few models trying to combine ILS and DTL events. We have the Lucas3 models, which is the model underlying DL-call, DL-parse, and SymPhy. And I'll start by describing the statistical implementation of the model, and then I will explain the actual model of evolution. To implement this model statistically, given a species tree, we first perform a birth-death process for duplication and losses on the species tree. The T is in bold here to stress the fact that DL-call and DL-parse have only DL events, while SymPhy has also transfer. At each duplication, the species tree is copied from that point onward and attached to the original species tree, where it may subject to further duplication, whether the species tree is truncated at each loss. This produces the so-called locus tree. The gene tree is then reconstructed by applying a multispecies coalescent process within the locus tree, with the caveat that only one gene copy can travel along the branch connecting a duplicated locus back to his originator. This means that all lineages of one particular gene copy must coalesce into one lineage before coalescing with its sister gene copy. So we use the so-called bounded coalescent process. Now let's talk about the model itself. Each species evolves as a panmimic population. Within a population, in each generation, each member may pass any of all its genes to any member of the next generation. Several alleles may be maintained in a population in any generation, so allele polymorphism is possible. Duplication and losses and transfer for SymPhy are the possible events that can shape gene history. When a gene duplication occurs, or a transfer for SymPhy, the new original gene copies evolve independently of each other, that is, the two loci are unlinked. Duplications and transfer in SymPhy must rejoin the original locus via the multispecies coalescent. The fact that the lineage of the duplicated individual joins via the multispecies coalescent, so after the duplication time, implies that the individual did not survive or not sample in the original locus. So the event is non-ancestral. For the duplication to be ancestral, the duplicated individual has both to survive and to be sampled. In this case, joining with the original locus would happen in a horizontal fashion, that is at the time of the duplication and not earlier as in the previous slide. Non-ancestral events are a reasonable consequence of the assumption that all loci evolve independently, since the probability of an ancestral unlinked duplication is vanishingly small. In this model, ILS cannot interfere with gene duplication, transfer, and loss in generating variation in gene copy number among species, since the duplicated and transferred genes are transmitted to all descendant species at the new locus, bearing gene losses. So some scenarios, as the one depicted in these slides or the next one, are not possible. The model of ILS and DTL interaction is the haplotype model. Again, I will first describe the statistical specification and then the model of evolution. Here we have the opposite then for the Lucas3 model. We first perform a multispecies coalescent process on the species tree, obtaining the so-called haplotype tree, and then the gene tree is obtained by performing a duplication loss birth-death process on the haplotype tree. Again, in this model, each species evolves as a panmimic population. Each species has a member that may pass any of all its genes to the member of the next generation. Allele polymorphism is possible, and duplication, transfer, and losses are the possible events that can shape gene history. The differences with the Lucas3 model are mainly three. So in this model we have that all loci evolve dependently, so no recombination is possible. We have the duplication are applied to gene lineage, so they are ancestral. And for a symmetrical reasoning of what done for the Lucas3 model and the non-ancestral duplication, here, ancestral duplication are a reasonable consequence of the assumption that all loci evolve dependently, since the probability of a non-ancestral linked duplication is vanishingly small. Finally, copy number hemiplasty is partially taken into account in this model, since duplicated and transferred genes do not have to be sorted in all species. But since they have to be sorted in the same species than the original copy, bearing gene losses, copy number hemiplasty is not fully allowed. Other methods exist that consider ILS and DTL, such as Notungen, etc., but they are rather reconciliation algorithms and not proper model of evolution, and they do not either properly take into account copy number hemiplasty, so because of time constraint I will not consider them here. I will now describe our model of gene family evolution, the Multilocus multispecies coalescent. Contrary to what done for the other two models, I will first describe the model itself and then give it an idea of the statistical implementation. So the first 3 points are common to the three models considered in this talk. So we have a panmimic population, the allele polymorphism is possible, and we have DTL as events. Then we have the CNH is allowed for duplication, transfer, and losses. This means that duplicated, transfer, and lost gene may fix in any subset, including none of all of the descendants. Then in our model, we have both unlinked duplication, as the Lucas3 model, and linked one, as the haplotype 3 model, where a combination can possibly break the dependency between the loci. Finally, transfer are always unlinked. Our new model has three major innovations. First of all, thanks to the incomplete coalescent, we can model copy number hemiplasty. Second, thanks to the coalescent rate process, we can calculate the rate of surviving DTL events. And finally, thanks to the linked coalescent, we can model partially linked loci. In the next slides, I will try to give an overview of these three processes. To do so, I need to introduce the concept of unilocus tree. So each duplication or transfer creates a new locus, and this new locus may be inherited by the descendants of the ancestral species in which it was created, and only by this. So for each newly created locus, we define a unilocus tree as the subtree of the species rooted at the time of the transfer or the duplication. So in our example, if we have a duplication at time t1, our unilocus tree will consider 3 species. For a duplication at time t2, our unilocus tree will only contain 1 species, the B species here. I will start by describing the complete coalescent. So in the complete coalescent, we do not require that all extant genes have coalesced to the most recent common ancestor by the time of the origination. So what we do is that we run a coalescent process that is stopped at the time of the origination, and it produces a haplotype forest. Then we choose randomly one of the 3 of the haplotype forest to be our haplotype tree. So here in this example, we have a unilocus tree on the left that has a duplication at time t and produces a unilocus tree on the right. Within this unilocus tree, we run a coalescent process that produces 2 trees, a blue and a green one. The blue one is chosen to be the haplotype tree. Since the blue tree has no branch in C, then we have copy number hemiplasty. Incomplete coalescent is easy to understand and simulate, but has deep implication. Indeed, although duplication occurs at constant rate, in other models they do not survive with constant probability. In other models, such as the Lucas3 model, the survival probability is constant in time and across lineages, but this is not true anymore in our model because of a gene copy number hemiplasty. This can be easily seen by considering the simple example of a duplication that occurs just before a speciation into 2 species leaves. In this case, we have that duplication can survive because it is observed in either of the descendant species and possibly in both. And since this probability is independent, the total survival probability is roughly twice of that of duplication which occur in a terminal branch of the tree. To calculate the surviving probability of an unique duplication, consider a duplication which occurs in a certain species. I consider the sample individual in the extant descendant of the species. Each of these individuals will have a single direct ancestor at the time of the duplication, who may or may not be distinct from each other. Then the duplication will be observed if and only if one of these ancestors is the duplicated individual. The probability that such a duplication is observed is proportional to expected number of lineages at the time of the duplication under multispecies coalescent. It is very difficult to obtain a closed formula for this number, but there is a simple way to simulate at the correct rate. For unlinked duplication, we run an incomplete coalescent in the unilocus tree, and then simulate event at the constant rate on the branches of the coalescent trees. The events are then considered to occur in the corresponding branches of the unilocus tree, allowing to simulate surviving duplication at the correct rate. On our example, we have on the left the unilocus tree on which we simulate coalescent trees and events on these coalescent trees. So in this case, we have 2 duplication square things. Then we forget the coalescent trees and we keep only the simulated events that are simulated at the correct rate. So let's discuss the last process, the linked coalescent, which is useful when a duplication produces a new locus which is physically close to the originating locus on the chromosome, in which case the evolution of the 2 loci may be dependent. So in the linked coalescent, we start by copying over to the new locus the haplotype forest of the new gigant locus, and this forms the persisting genealogy which can be formed. Then we run the incomplete coalescent, in which the lineage can coalesce with each other, as usual, or with the pre-existing genealogy. In this way, the genealogy of the new locus depends on the genealogy of the existing locus, and in absence of recombination, the two will be identical. But then, to model the effect of recombination, we have that lineages in the new locus, which are coalesced with the copied genealogy, can coalesce from it, and this represents a recombination event. In contrast, lineages in the new locus, which coalesced with each other, cannot coalesce. Then, by varying the rates at which recombination events occur, we can model any level of dependency between loci. Aiming at assessing the impact of CNH on gene evolution, we simulated gene trees on the fungal species tree used by Rasmussen and Kellis, using 3 different duplication and loss rates, where duplication and loss rates are considered equal here. 3 effective population sizes, and 0.9 year per generation, which includes similar parameters of those considered by Rasmussen and Kellis in their paper. And to be similar to their scenario, we also consider all duplication to be unlinked and no transfer. For each set of parameters, we ran 500 simulations and calculated the proportion of unilocus tree for which the haplotype forest contain more than one haplotype tree. So this means that CNH occur in these cases. From this figure, we can see that for this dataset, CNH is far from negligible, and as expected, its impact is higher when the effective population size is large, ranging from a bit less than 15% to more than 50% of the time, while it stays constant when varying the duplication and loss rates. And this comfort us a lot in the fact that modeling CNH may be very relevant for some biological systems. To conclude, the multilocus multispecies coalescent permits to account for copy number hemiplasty. Also, even though it does not simulate a fully recombination, it can take its effect on events into account in a natural way. Finally, recognize the fact that the realized rates of duplications, transfer, and losses become inhomogeneous in a case where ILS is at work. With this new model at hand, we can use the simulated gene trees, for instance, to compare the accuracy of current reconciliation algorithms in a neutral setting, or the accuracy of current ILS-aware Soviet tree reconstruction algorithms, such as Astral, in more complex scenario that includes CNH. This simulation can also be used to ascertain the relative importance of this population-level processes to microevolutionary ones. Finally, we are currently working on a reconciliation method that is based on this new model. And the last slide to tell you that if you're looking for a new wave of publishing, then PC Mathematical and Computational Biology is waiting for your submission. Thank you so much for your attention.
Okay, can you hear me? Okay.
Yes.
Okay, now we are going to face the biggest technical challenge of the meeting, that is doing an Skype in an area with a low internet connection. So let's try that. Okay. So I will share now this Skype, and we are going to make a phone call. Can you hear it, Rosa?
Yes. Yes, I can.
Hello, Céline. Can you hear me?
Yes, I do.
Okay. And Rosa, you can hear Céline?
Yes, very low, but I can hear her.
Okay. I don't know if I can improve that. So let me check.
I don't know. I will try. I will ask the first question. Okay, and let's
Yes.
we could something. So. Okay, so far, there is any question that you would like to ask? We're waiting, setting for some questions.
Don't be shy. Feel free to ask any questions you may have for Céline. Otherwise, go ahead, Jesus.
The moment. In case there is no question, we are waiting for some. What we could do is we are going to let the Slack channel open. Maybe you will be able to reply them afterwards.
Yeah, on written form, yeah, no problem.
That's fantastic, Céline. So thank you very much for staying there, and yeah, I hope that you enjoy your time.
No, let's give people perhaps 30 seconds to
wait
ask.
a second. Rosa is telling me something.
I was just saying, Jesus, let's wait a few seconds in case someone wants to ask something.
Okay.
Yes, we have a question in Slack. Can you read it to Céline?
I will read it. The question from Pascal is not serious. So how did you choose duplication rates in your simulation?
So what we wanted to do is to show that on a similar setting that what was used in the Rasmussen and Kellis in 2012 paper, we were going to expect pretty much hemiplasty. So we took some rates that were on the range of the rates that they used. And they were also used in a few other papers from Tandy Warner, Mirarab, Nackley, that they were trying to test some supertree methods such as Astral and Astral Dupe, Astral Pro. And they were using also the fungi tree. So this is how we chose them. So there are 3 different rates of duplication and loss that are equal that are on the slide. That are ranging from 10 minus 10 and 10 to minus 9. And they were, similar to Rasmus and Callis, and the same that other people used. So they are rates that are biologically relevant.
Okay, Céline, thank you very much. I don't know, Rosa.
You're welcome.
There are more questions, Jesus.
Okay,
Have a look in
let's go for the second one by Jacob Winter. Is ILS at all detectable when looking in ancient phylogeny, something like 500 million years or older?
Yes, you can definitely find some signal. Then the problem is always that when you start adding different events, then they can be confounding processes. But it's not the lack of signal that create the problem, but the fact that there's lots of things that can go on, such as gene flow and ILS, and then these are the confounding things. But if the signal is strong enough, even if it's ancient, then you will find that that node that has been confounded by this kind of events will be confounded still in our phylogeny that we reconstruct now.
Okay. Thank you very much. They thank you on the Slack channel.
And I have a question as well.
Okay, Rosa, you can hear directly Rosa, or maybe I will transfer the question. Make the question, Rosa, please.
Yeah, can you discern the difference between missing data because you have not sampled a gene and real gene loss?
Have you been able to hear that?
No, I cannot hear at all.
Okay. Have you been able? Can you repeat it again, Rosa?
Can you discern the difference between missing data because you, for example, are working with a transcriptome and you have not sampled a gene and gene loss?
Okay, can you discern between missing data, maybe because you have been working with a transcriptome, or something that has been really lost?
No, like a true gene loss.
Yeah, this is difficult, and I don't know. I don't think we can. If it's not there and it should be there, then the only thing that I can think of is a process such that if it was there, we would put it there, and then maybe trying to reconstruct some ancestral sequence and then see if we can blast and see if we didn't miss it, but not with this kind of method.
Yeah, sorry, maybe I didn't formulate well the question, but I guess that Rosa will write it down properly, because
that's okay. It was great. Thank you.
Thank you very much. So I think for the time we should move on. Thank you very much, Céline. Okay, it has been a pleasure.
Thank you so much.
Thank you.
Enjoy the conference, and thank you again for the invitation.
Thanks to you. Bye.
Thank you, Céline. Thanks a lot. It was a very stimulating talk, definitely.